Hello, this is a review for Unit 2 Diabetes for the PLTW PBS course. Our first review question was talking about the relationship of insulin receptors and then also the GLUT4 transporter. If we're looking at our picture to the right, you'll notice that the insulin is going to be represented by those yellow dots. Insulin, remember, is going to be a hormone and it helps to lower blood sugar. How does it actually work though? What happens is that the GLUT4 transport protein needs to be open. In order for that to happen, the insulin molecules right here are going to bind to the receptor. Note that the insulin and the receptor both have specific shapes. That's because they're both examples of proteins. When the insulin binds to the receptor, if you focus your attention then on the GLUT4, that transport protein is actually able to help move glucose across the membrane. When glucose is able to get into the membrane, then cellular respiration can take place. Glucose plus oxygen is used by the mitochondria to produce a usable form of energy known as ATP. So we have for step 1, the hormone insulin binds to the specifically shaped protein receptor. This causes the GLUT4 transport protein to open, which allows glucose to enter the cell. When glucose enters the cell, this means that your blood sugar level is going to go down. The cell can then use that energy for cellular respiration. Now we have the fact that there are two types of diabetes, type 1 and type 2. Remember, type 1 diabetes is largely going to be considered a genetic problem, whereas type 2 is largely caused by lifestyle. Type 1 diabetes is going to be when you do not produce enough insulin. In a couple of minutes, we'll be looking at the graphs that you will have to analyze in order to diagnose a patient. People with type 1 diabetes cannot make ATP as well as somebody else, and that's just really because there's a lack of glucose getting into the cell, so that mitochondria is not able to undergo as much cellular respiration. How can you help to treat type 1 diabetes? You're going to have an insulin pump or insulin injections. Someone's going to have to make sure that they check their blood sugar regularly. And based on what their blood sugar is, that'll determine what foods they should eat and then how much insulin they should intake. Type 2 diabetes, once again, is going to be lifestyle dependent. The cell receptors do not recognize the insulin. So remember, they're both examples of proteins. Proteins have a specific shape, which are going to determine their specific function. The two are not really working like a lock and key. Once again, ATP cannot be adequately produced by cellular respiration because, once again, the end result is that glucose is not effectively getting into the cell. In order to treat this, you also can undergo insulin injections in a pump. And another key factor in this is going to be lifestyle changes. Modifications to your diet, such as lowering the amount of sugar that you're intaking, exercising, and weight loss. How would you know potentially if you do have type 1 or type 2 diabetes? What things could you be on the lookout for? Here we have frequent urination, weight loss, thirst, fatigue, and blurred vision. We'll go over a couple of reasons why those things happen if you have diabetes, depending upon if you're hyperglycemic or hypoglycemic in a minute. Looking here, this is that graph that I was talking about. You are able to generate this graph because of a glucose tolerance test. Looking at the graph, there are going to be two lines on there. There is the insulin production as well as the glucose production over an extended period of time. Looking here, you'll note that there are different values. The 200, if it goes above that, that's going to indicate that the person has diabetes. That is not how you really can determine, though, if the person has type 1 or type 2. How can you determine that? You have to look at their insulin value. If their insulin value stays very low, under 100, that person is going to have type 1 diabetes. So looking here, this insulin value is under 100. And since it's under 100, we're going to say they have type 1 diabetes. For the reasoning or your explanation, you're going to write down that their sugar level is above 200 milligrams per deciliter. And then additionally, they have low insulin. Moving on to the next graph, once again, I'm always going to go and I'm going to look at what the value is. Note here, again, I make that line at the 200 so I can recognize if glucose goes above that or stays below. I notice that glucose in this case stays below the 200 line, so they are not diabetic. It actually is also below the 140 line. 140 would indicate that the person is pre-diabetic. If you look at insulin, there's a spike in insulin, but as that insulin increases, that's going to cause that glucose to decrease. This person is actually maintaining homeostasis. They're in dynamic equilibrium, more or less a steady state. This person is normal. They don't have diabetes. They're also not pre-diabetic. How are you able to tell this? Again, insulin's released. It lowers the blood sugar level. Remember, that's an example of a feedback mechanism, and that sugar is less than 200. Our final graph, again, we look for that 200 line to indicate if the person has exceeded that. This person has in fact exceeded 200, so they are going to be diabetic. The other thing we look at is insulin. What you'll notice here is that they actually are able to make insulin. Their number goes well above that 100 milligrams per deciliter. Because of that, they are going to be diagnosed as type 2 diabetic. So insulin is released, but that feedback mechanism is not working properly. Once again, that is going to be because there is a problem with the lock and key mechanism of the insulin and then also the insulin receptor. They're not fitting together properly. So even though they're making insulin, glucose is not getting into the cell. Here, they're giving you a situation. It makes note that this person has a blood sugar level of 160. That is going to be elevated. Is it very elevated? No, but it's still above that 140, which is when someone starts to be considered pre-diabetic. So looking here, patient should apply heat. Heat's going to have no effect on your blood sugar level, so you can cross that one off. Next one says patient should take insulin. Remember, insulin is going to lower your blood glucose level. So that could be an answer choice. Next one says the patient should take glucagon. Glucagon actually raises your blood glucose level. In this case, we want to lower it. They have high blood sugar, so you're going to cross that off. The patient should drink orange juice. Orange juice has a lot of sugar in it. If you were to drink orange juice, you would actually have a spike in your blood sugar level. The patient should go to the ER. 160, while elevated, is not really going to be causing you to have to go to the emergency room. You can most likely regulate that on your own by taking your insulin. Last one says the patient should recheck their blood glucose level in 10 minutes. That is a good idea because after you take your insulin, you want to make sure that your blood glucose level actually does go down back within normal limits. In terms of lifestyle suggestions for someone who has type 2 diabetes, this has to do with diet, lower the amount of sugar that you're intaking daily. The other thing that you're going to want to do is lose weight. And then finally, exercise. And remember, these do not have to be big changes. It could be things like increasing the number of steps you're doing per day. Weight loss slow and steady does not need to be a crash diet, and just making sure that you're making healthy choices. Over on the right, we have an example of a feedback mechanism. Now, there are two types of feedback mechanisms. The first feedback mechanism is going to be one that is referred to as a positive feedback mechanism. Let's say that you are in labor, for example. What happens is that your body increases the production of a hormone. When your body produces this hormone, this is going to cause an increase in contractions. When you have more contractions, what happens is that then you produce more of that same hormone that you initially produced, which in turn leads to more contractions. That is positive because you have a plus, a plus, a plus, a plus. The two of them are related to one another in a direct relationship. The next type that you can have is going to be a negative feedback. Negative feedback has the opposite actions. If we look here, we have a classic example of blood sugar levels being regulated. Here, this would be an example. If you eat something, you would get a spike in your blood glucose level. Then you have a response. That response is telling your body, hey, that blood sugar level needs to go down. Insulin gets produced. Insulin is going to lower your blood sugar level. Notice here, we have a positive followed by a negative. Your blood sugar level goes back to normal. Feedback mechanisms, just that last little example. We can be talking about regulating your blood sugar level as one example. Regulating your body temperature would be another example. If you do fail to maintain homeostasis, then you would be diabetic. People who are diabetic can't regulate or control their blood sugar level. Someone who's diabetic could be suffering from hyperglycemia or hypoglycemia. Over here, this is just a general overview of what the major macromolecules are. We have proteins are made up of amino acids. Remember, those are going to be critical to pretty much everything that's happening in your body. Some examples are hemoglobin. Hemoglobin is responsible for transporting oxygen around your body. Then we have enzymes. Enzymes are going to be responsible for speeding up chemical reactions. We have antibodies, which are produced by white blood cells in order to help you fight infection. Lipids are going to be responsible for energy, obviously. They have a lot of chemical bonds. Next up, we have hormones. Hormones are responsible for things like insulin. They're responsible for testosterone, estrogen. Your cell membrane is made up of a phosphobilipid layer. Insulation in terms of keeping you warm. Carbohydrates are looking at energy. And then nucleic acids are really just your genetic information. Here are just two terms to review really quick. Hydrolysis is going to be digestion, and that's when you add water. And then we have synthesis. Synthesis means to build or make. And then dehydration just means to remove the water. So you're building or making things by removing two H's and an O. And when those come together, then you've got water as a byproduct. Over here, we have some chemical testing. You should be familiar with it. Chemical testing for Benedict's, that's going to test for glucose. And this orange color would be a positive. Iodine is going to test for starch. Amber is actually a negative. If it was a positive, it would be represented by blue-black. Paper bag is going to test for a lipid. Remember, lipid is really just a fancy word for a fat. Transparent is going to be a positive test, meaning that it's shiny, kind of see-through. And then finally, biuret is going to be an example of testing for a protein. When you're testing for a protein, that is going to be positive if it is purple. Looking through here, food A would have glucose, lipid, and protein. Again, because each one of those indicators, the color of them indicates positive for the presence of that particular macromolecule. First up, we have hypoglycemia. You definitely need to know the effects for our final, so make sure that you review that. Hypo, I always think low blood sugar. And again, that's what's surrounding the cell. So if we were to have an example of a cell, there would be a certain number of glucose molecules on the inside, and then there'd be a certain amount of glucose molecules on the outside. What you'll notice is that the number of glucose molecules on the outside is much less than the number of glucose molecules on the inside. So therefore, the outside is considered high water concentration. Water, in this case, would diffuse into the cell. Effects of this: shaking, sweating, anxious, irritable, hungry, fast heart rate, fatigue. Think about it. These people have low blood sugar. They don't have enough sugar in order to supply their cells. And because of that, they can't make ATP. If they don't make ATP, they're going to feel tired, irritable, all those other things. Next up, we have hyperglycemic. Hyperglycemic, I think hyper, lots of sugar. This is actually the exact opposite of that hypo situation. If I have this situation, I've got a lot of glucose on the outside and just a couple of glucose molecules on the inside. That means that the water, the blood that's in the water surrounding the cell, is going to be considered a relatively low concentration. Because of that, water is diffusing out of the cell. If your cells don't have enough water, these effects should make a lot of sense, right? You're going to be thirsty. Your cells are dehydrated. And because your cells are dehydrated, that's because water has been moving out of your cells and going into your blood. The blood then gets filtered by your kidneys, and you're going to have to urinate more frequently than someone who's not experiencing hyperglycemia. Dry skin, hunger, drowsiness, nausea. So once again, you have to know the difference between hypo and hyper, and you should also know some of the effects or symptoms that someone would be exhibiting if they have those conditions. Isotonic just means equal concentrations on both, so therefore that person is in homeostasis or in dynamic equilibrium. Looking at this picture here, once again, they want to know hyperglycemic, hypo, or isotonic. The outside is supposed to represent the blood. This here is the actual cell. If I look there, there's so many glucose molecules around that cell. If there's a lot of glucose, right, maybe you want to think, hey, that person has a lot of sugar. A lot of sugar, they're going to be really hyper. So the blood itself is going to be hyperglycemic. And again, that has to do with that sugar concentration. So hyperglycemic, and when we're talking about something that's hyperglycemic, we're talking about a high blood sugar concentration. Which way will the water move? Water is always going to move from high to low. We're not looking at the sugar content, we're looking at the water content. So here, this is actually inside of the cell, has a high water concentration. Outside of the cell is going to be considered the low. Because of that, that water is going to diffuse out of the cell. So water would diffuse out because it's moving or diffusing from that high to low concentration. And the last thing is, what is the name of the test that diabetics are using in order to determine what their blood sugar level is? And that's going to be the hemoglobin A1C test. That shows you your average blood sugar concentrations.